Hello, I welcome you all in this module on maturation of kerogen, the biological and thermal effects. Now, after studying this module, you shall be able to understand types of organic matter, their transformation to kerogen and classification of kerogen on the basis of hydrogen is to carbon ratio. You will be able to learn about different stages of transformation of organic matter, causative agents, that is biogenic and thermal, behind this transformation process and relative roles of agents at different transformation stages. You will also be able to know about different parameters and indices. Those are helpful in assessing maturity of sediment and the criteria which will follow to categorize any sediment body as an effective source rock. Now, potential hydrocarbon source beds are formed dominantly by thermal maturation of organic matters present within sediment under the combined effects of thermal, kinetic energy and geological time as sediments undergo burial. In diagenesis, biogenic decay and abiogenic reactions suffered by organic matters release methane, carbon dioxide, water and leave a complex hydrocarbon compound within sediments that is referred to as kerogen, and that is made up of carbon, hydrogen and oxygen with minor amounts of nitrogen and sulfur. It is differentiated from bitumen by its insolubility in normal organic solvents like carbon disulfide. The transformation of organic matter to kerogen takes place right from shallow burial with near surface temperature up to a depth of 1 kilometer, that is about 50 degree centigrade temperature. In a steadily subsiding sedimentary basin, the transformation of organic matter to kerogen and its subsequent maturation to oil and gas happens under three different stages, that is diagenesis, catagenesis and metagenesis. Now, maturation is a combination of processes through which complex biological molecules created by living organisms are first converted into simple biomolecule and finally cooked to give petroleum. In the early stages of alteration, that is what is termed the diagenesis, an intermediate form of organic matter called kerogen is formed. Now, dehydration, that is release of water, and decarboxylation, that is the release of carbon dioxide, are the two main reactions at this stage, dominantly under the influence of bacterial action. The EHPH condition at the sediment water interface principally guides bacterial action on organic matters, while free sulfur is released from sulfate ion and with removal of oxygen under the action of desulfovibrio bacteria in reduced condition below water table, oxidation of free sulfur by the bacteria Thiobacillus in an oxygenated condition result in the formation of sulfate ions. In general, bacterial action at first oxidize organic matters and release water, carbon dioxide, nitrates and phosphates. Reduction reactions follow which turn nitrates into nitrites and sulfates into hydrogen sulfide and ammonia. Organic matters are diverse and complex in constitution and made up of proteins, carbohydrates, lipids and lignin, with proteins being most unstable and lignin most stable. Microbial enzymes decompose these compounds and form biomonomers, that is proteins transform into amino acids, carbohydrates to sugar and lipids to glycerol and fatty acids. Cellulose gets converted to methane and carbon dioxide. In fact, methane constitutes a major product of bacterial decay. All these changes happen in top few meters of the sediment column, and further with increase in temperature and time with respect to time, the chemical reaction starts dominating over bacterial action. Net outcome of all diagenetic reactions is the reduction of oxygen content within the organic matters, keeping carbon and the hydrogen ratio intact. As burial continues in deep subsurface at catagenetic stage, fluid phase is released from kerogen, that is oil and then gas, which further decrease the hydrogen is to carbon ratio in the kerogen. A fluid phase high in hydrogen is released and a residue that is rich in carbon that results. A still higher temperature causes condition that we call the metagenetic stage. Almost near to metamorphism, the last hydrocarbon, that is methane, is released, leaving behind extremely carbon rich graphite residues. Although this represents the general pathway through which organic matter and kerogen matures, examples are available where thermally immature Cambrian source beds about 500 million years old are exposed in outcrop in southern Sweden and gas generating overmature tertiary source beds belonging to Miocene shells encountered along the west flank of the Red Sea in Ethiopia. While in the first case the time elapsed for the Cambrian shells provides the required compensation for the thermal effect, the high thermal gradient in the southern Red Sea, similar to the high gradients found today, is identified as the causative reason in the second case. So, the hypothesis about the thermal maturation histories of potential source beds must be used very carefully. In geochemical modeling of hydrocarbon source rocks, geologists widen their range of input data and use data from the fields of coal petrography also, such as the vitrinite reflection data or coloration of spores or chitinous microfossils if they are available. Detailed chemical analysis of oils and source beds for specific compounds also add to our understanding of the thermal histories of rocks and basins. A reliable interpretation of source bed maturation is possible only after all data they are collected and processed. Now, we discuss that what are the different types of organic matters and kerogen that we encounter. Now, depending on whether it is coming from terrestrial plant or it is coming from the aquatic, that is plant or animal origin, organic matters are broadly classified as humic and sapropelic. In character, humic organic matters are largely carbohydrate lignin in composition with protein in subordinate amount, relatively rich in carbon and nitrogen and poor in hydrogen. Thermal alteration of humic organic matter results in humic acids rich in oxygen and poor in hydrogen. These organic matters are gas prone and poor in liquid volatile product yield. In contrast, sapropelic organic matters are of aquatic, either they may be marine or lacustrine, made up of structureless vegetable slime or polymerized lipid and protein. Spores, pollens, cuticles, alginides, all these with high hydrogen content constitute this organic matter, and thermal alteration release liquid volatile products, that is oil, from these organic matters. Differentiation between these two types of organic matters can be done taking into consideration their hydrogen is to carbon contents. The hydrogen is to carbon content in humic organic matter is always less than 0.8. In mixed humic and sapropelic organic matter, this hydrogen is to carbon ratio that will be between 0.8 and 1, and in sapropelic organic matter it is above 1. So, at diagenetic stage, organic matters transform under bacterial attack into a thermally reactive organic complex, that is kerogen, with release of methane, carbon dioxide and water. Now, you see there is in this diagram the different kinds of organic matters, that is algal, which we call the type 1, where you get the spores, pollens, they are shown. Then the type 2, that is the mixed marine, then the type 3, that is the terrestrial woody, and the inert, that what we call the barren, that is type 4. All these organic matters, how they are, that is being shown. You can see that some of the structures of the organic matter that is shown in the figure in the inset within the figure, and then we can have a algal type, that is the type 1, we can have a mixed marine type, that is type 2, coaly type, that is type 3, and the inert type, that is the type 4. Now, microscopically, kerogen can be seen as yellow-orange to brown-black particles or amorphous material. Since kerogens originate from different kinds of living organisms with different kinds and proportions of biological molecules, their chemical compositions vary widely and result in formation of hydrocarbons of different types and amounts. Geologists have found it convenient to group kerogens into 4 fundamentally different classes ranging from type 1 to type 4. Now, type 1 kerogen, it is derived from sapropelic organic matter, principally constituted of the high grade algal remains which are lacustrine with high hydrogen is to carbon ratio, that is 1.6 to 1.8, and on maturation it yields crude oil. It represents the most petroleum generating kerogen type. Now, type 2 kerogen consists mostly of amorphous material, both continental and aquatic in origin, and derived from the bacterial and mechanical breakdown of a mixture of marine, one-celled plants and animals. Algal tissues, spores, cuticles, pollens, they also contribute. This organic type has H:C ratio about 1.4 and oil prone, but yields more natural gas than type 1. Now, type 3 kerogen, here the hydrogen is to carbon ratio is 1 or less, is derived from humic organic matters and they are of terrestrial woody origin from higher land plants and sometimes known as coally-kerogen, equivalent to vitrinite macerals in coal. The humic material in type 3 kerogen has a low capacity to form oil and yields mostly natural gas. Type 4 kerogen, where the hydrogen is to carbon ratio is 0.4 or less, consists mostly of inert particles that have already been highly oxidized, recycled and altered from burial like altered before burial like charcoal. It is the rarest kerogen type and has practically no ability to yield any kind of hydrocarbon such as oil or gas. The chemistry of crude oil can also be linked to kerogen type and the original organic matter. Usually land derived non-marine organic matter deposited near continental drainage areas that are normally type 3 coally-kerogen and will form mostly gas, and any oil formed will be of low sulfur, paraffin to paraffinic naphthenic crude oil. Marine organic matter, particularly protein rich types derived from marine animals, that is the type 1 or type 2 mixed marine kerogen, tends to yield high sulfur aromatic intermediate crude. Now, this is a diagram which is very popularly known as the Van Crevenen diagram, where you can see there are 2 axes, that is the one axis that is showing the atomic oxygen is to carbon ratio and the other axis is the atomic hydrogen is to carbon ratio. Now, all the 4 kerogen type, that is type 1, type 2, type 3 and type 4, their evolutionary path that is being shown in the diagram, right as they are going through the biological and the thermal maturation process. What we can see that above this diagram we have superimposed the 2 more the maturation index. One is vitrinite reflectance, another is the thermal alteration index. So, one side the VR indicates the vitrinite reflectance and TAI, that is indicating the thermal alteration index. It is clearly shown that the type 1 and type 2, as they are evolving through time and through the biological and the thermal process, they have the potential to yield oil and gas, where the more oil potentiality is that of type 1, but the type 2 also yield oil, but the gas potentiality capability is more than that of type 1 within type 2. The type 3 kerogen, that is more prone to give you coal, and type 4 that is already oxygenated, so they have very weak potential to give any kind of hydrocarbon. Now, all these what we have discussed, that how these different type of kerogen, that is the humic, sapropelic, and then we categorize them with respect to hydrogen is to carbon, the oxygen is to carbon, and how they are maturing through the effect of the biological and thermal process. These processes what cause the kerogen to be matured, that is also very important for us to understand. Now, biological transformation of organic matter to kerogen and chemical transformation of thermally reactive kerogen give rise to the generation of petroleum. Bacterial processes remain mostly effective the initial breakdown of organic matters, while cooking of transformed organic matter, that is kerogen, is guided by the thermal process. A combination of negative redox potential and alkaline pH condition, that is between 7.5 and 8.4, favors liberation of petroleum. In isolated bottom condition, where generation of H2S turns bottom condition acidic with pH lower than 7, hydrocarbons are retained in the kerogen and not liberated. Both temperature and pressure increases as organic matters get buried. Transformation of organic matters is independent of pressure. In fact, high pressure may counter rate of decomposition. So, the main factor what is important for the transformation, that is temperature and time. It is not pressure. Now, temperature plays pivotal role in organic matter transformation. Higher molecular weight organic matters transform into simpler compounds by thermal cracking. Generation of petroleum is in fact a rate controlled thermocatalytic process. In addition to temperature, time spent by organic matter at different temperatures also play a key role. Since thermal decomposition of organic matter is a temperature dependent chemical decomposition, it is expressed by a simple equation that is being offered, and we call it the Arrhenius equation. The Arrhenius equation, it is essentially a temperature dependent chemical reaction where you see that there is a pre-exponential factor dependent on temperature, there is activation energy of reaction and the universal gas constant, and temperature is shown in the form of Kelvin. And according to this equation, reaction rate doubles in every 10 degree increase in temperature. Since generation of petroleum is a chemically reactive process, the amount of petroleum generated will vary linearly with time and exponentially with temperature. Now, we further elaborate the effect of temperature and time, and this diagram is very explicit to show that what is the effect of the temperature and time. Now, petroleum is generated when kerogen is subjected to the increased temperatures that accompany the sediment burial. Now, in this diagram, it is shown that the increase of temperature with respect to depth and the generation of the hydrocarbon, that basically is divided into 4 different zones. That is the bacterial zone, then the dry zone, then the wet gas zone, and then the dry gas zone. The best oil production, that is the oil zone, that is refined between 60 degree and 175 degree temperature, what we normally refer to as the oil window. So, this particular temperature, that is 60 to 175 degree, that basically is the major zone where we should expect the formation of oil because of the maturation of the kerogen. More than that, in the shallow part, what we see that the gas generation, that is basically because of the bacterial action, and more than 175 degree centigrade in the higher temperature, whatever the gas generation, that is the dry gas we are referring here, that is because of the thermocatalytic action. The alteration of kerogen thus is similar to the thermal cracking reactions. Large kerogen molecules decompose upon heating to yield smaller molecules of petroleum, and these reactions usually require temperature greater than 60 degree centigrade. At lower temperatures during early diagenesis, natural gas, which is because of the biogenic methane and marsh gas production, is generated through the action of the microorganisms that live near the Earth's surface. Vast quantities of biogenic methane are probably generated, but most of these will not be encountered in a trap and will be lost in the atmosphere. The temperature range between about 60 degree to 175 degree, as I have already discussed, that is what is called the oil window. This is the principal zone of oil formation. It begins at burial depth of 1 to 2 km and ends at a burial depth of 3 to 4 km. In most areas, depending on the factors such as geothermal gradient. The first oil generated is heavy and tends to be richest in aromatic and NSO compounds. As burial and temperature increases, oil becomes lighter and more paraffinic. At temperatures much above 175 degrees centigrade, the generation of liquid petroleum ceases and gas formation becomes dominant. When formation temperature exceeds 225 degrees centigrade, most kerogen has used up its petroleum generating capacity. Along with a series of chemical reactions, time also play a very key role in the petroleum generation process. Young tertiary age rocks need to be deeply buried and should have the high geothermal gradients in order to generate significant amounts of petroleum. Although generation, migration and entrapment have been documented in rocks as young as 1 to 1.5 million years old, major petroleum accumulations have not been identified in rocks younger than 10 million years. On the other hand, some old Paleozoic and Mesozoic source rocks might not have been buried very deeply, that is buried only up to the uppermost level of the oil window, but have still generated petroleum because of the time factor. However, in most petroleum occurrences, temperature appears to be a more significant factor compared to time. Now, the chemical law of kinetics that is expressed in the Arrhenius equation states that the rate of any chemical reaction is related with temperature and time. It is estimated that the reaction rate doubles with every 10 degrees centigrade in temperature, as I have already discussed. So, two commonly used indices of maturation that we study to understand the maturation of organic matter is the time temperature index TTI and level of organic maturation, that is LOM. While TTI integrates temperature increase with time spent in each temperature interval for an incremental increase of 10 degrees centigrade, in course of sediment burial, the LOM scale is framed with the consideration that reaction rate doubles in every 10 degree increase in temperature. In LOM scale, oil generation occurs between 7 and 13 and prominent gas generation occurs between 13 and 18. With increase in depth and temperature, kerogens undergo progressive carbonization with reduction in hydrogen to carbon content. In fact, the thermal maturation results in release of a fluid product rich in hydrogen, industrially known as oil and gas, leaving behind a residue very rich in carbon. Hence, hydrogen content or carbon content of a source rock is considered a good proxy for its degree of maturation. Since hydrogen content of sediments may also include the hydrogen that is contained with pore waters or in combination with clay minerals, the carbon ratio is considered a good indicator for degree of maturation, that the carbon ratio is the dry ash-free fixed carbon divided by the fixed carbon plus volatile carbon together into 100. The fixed and volatile carbon values in the equation are determined by pyrolysis. Now, for sediments other than coal, that is shale, we resort to some other kind of parameter, that is hydrogen index, and estimation of volatile, that is the retortable carbon. Now, what is the retortable carbon? The retortable carbon, that is the organic acids, a major constituent of organic matter that react with potash to result potash products. This reaction is named as saponification, and in course of progressive maturation of kerogen, saponifiable components of organic matter get changed to non-saponifiable products, and this degree of transformation from saponifiable to non-saponifiable products is referred to as the hydrogen index. So, hydrogen index is non-saponifiable compound divided by saponifiable compound into 100, and sediments with hydrogen index 100 indicates complete conversion and over matured, and all saponifiable compounds transformed into non-saponifiable compounds. Commonly, sediments with hydrogen index above 90 are considered as suitable for prolific oil generation, and indices less than 40 are indicative of immature, recently deposited organic matters without any significant maturation. Another index of maturation that we estimate is the ratio between the retortable carbon, the carbon that can be volatilized at 500 degrees centigrade in an inert atmosphere, and total organic carbon. Total organic carbon of any sample is measured first by removing the inorganic carbon present in the form of carbonates by acid treatment, and then by measuring the carbon dioxide combustion product of the sample at about 1000 degrees centigrade. Hydrocarbon source sediments yield values between 0.4 and 0.5, at cases even higher, that is 0.6 or more. Non-productive or barren shells give values 0.1 or less. That what these parameters actually measure is the volatile content present within any sample. With progressive maturation, as volatiles are lost, accordingly the hydrogen is to carbon content of organic matters also reduces. However, for peak hydrocarbon generation from organic matters, elemental carbon in organic matter should not be more than 80 to 90% and hydrogen content should not be less than 7%. Preferably it should be above 10% by weight. It is suggested that the quantity of hydrocarbon generated from any sample generally maintain a linear relationship with the organic matter content of that sample, which is termed the oil quantity factor. This diagram shows what is the oil quantity factor, that is the organic carbon content and the oil quantity factor. You can see it is a linear relationship, but when you plot the oil quantity factor with respect to hydrogen is to carbon atomic ratio, it is not it is a non-linear relationship. So, amount of volatilized carbon in organic matters is identified by pyrolysis. In pyrolysis, what we do, the organic matter is heated in a stream of helium in an inert atmosphere. Temperature is gradually raised in a controlled manner at a carefully measured rate in Rockefeller pyrolyzer, and expelled volatiles are recorded in a hydrogen flame ionization detector. At 200 to 300 degrees centigrade temperature, free hydrocarbons present in sample get volatilized. This is referred to as the S1 peak in the pyrolysis curve. With increase in temperature, hydrocarbons are expelled from kerogen, which is designated as S2, that also we call the Tmax in the pyrolysis curve. On further heating up to a temperature around 390 degrees centigrade, expulsion of CO2 is marked by the pyrolysis curve, what we refer to as the S3. Pyrolysis continues up to 800 to 900 degrees centigrade. The readings, that is S1, S2 and S3, are used for estimation of source rock maturation. The ratio S1 by S1 plus S2, this is referred to as the production index and is used to estimate the amount of hydrocarbon generated with respect to the total amount of hydrocarbon capable of being generated from that organic matter. The ratio S2 by S1 plus S2 referred to as the hydrogen index, that correlates directly with the H/C of the organic matter. The area of S3 denotes the CO2 that evolved from the pyrolysis, and the ratio S3 by S1 by S2, that the oxygen index correlates with the oxygen is to carbon ratio of the kerogen type. Now, this diagram is basically the pyrolysis diagram. You can see that it is the three different peaks, that is S1, S2 and S3, that what I was just discussing, that what is the meaning of the three curves that we get, the three peaks in fact, what is the meaning of three peaks, what we discussed just now. Now, another method that is in use for maturation study is gas chromatography. With increase in burial, the depth of organic matter and temperature, there is more evolution of N-alkanes. A correlation is established between changing pattern of chromatograph and distribution of N-alkanes. In immature source rocks, N-alkanes show a broad whaleback with a bimodal distribution of unresolved components. With increase in temperature and maturation of organic matter, the whaleback submerges and unsolved components define a single peak. Broad low whaleback devoid of spike denotes the degraded oil, and this diagram clearly shows that what is the meaning of that. The first one at the top, you can see there are two peaks and there is a broad whaleback at the base. Then you can see that whaleback is diminishing, and the two broad peaks, they are also merging, and there is a general high. But as it is more evolving, and then you see that there is a clear peak, that one peak, and with very low whaleback, that basically shows that what is the character when it reaches to the oil window or when it gets the over matures. Now we come on to some optical thermometer that we use to identify the maturation of organic matter. The spore coloration index SCI and vitrinite reflectance. These are the two major optical coloration index or the optical parameter that we measure to understand the kerogen maturation. During thermal maturation, dispersed organic matters including spores within sediments, they change their color from yellow to brown to black. The change in color depends not only on rise in temperature but also on the duration of time through which the organic matter sustains that temperature, that is the geothermal history. The changes that happen in organic matter is time dependent, unidirectional, progressive and irreversible, and that is why we can use it as a criteria. Keeping this in view, a 10-point spore coloration scale, that is SCI, that has been devised. Organic matters of animal origin are also used for the same purpose, provided they are chitinous or carbonaceous. That is, conodonts display a comparable progressive color index as we get from the spore coloration index. Vitrinite reflectance in oil immersion with the use of reflectance microscope has been in regular use in source rock maturation study. Now, vitrinite measurer records maturation in its reflectance value, which is dependent both on time and temperature. Measurement of this value of representative samples with different mixed carbon contents allow reconstruction of maturation gradient curve, the gradient of which is dependent on geothermal gradient and the sedimentation rate. Now, taking into consideration both SCI and VR, that is spore coloration index and vitrinite reflectance, an empirical relationship is established between hydrocarbon generation and these paleothermometers. Crude oil generation is noticed between the reflectance values, that is 1.5 and 3, which matches with spore coloration, that is dark yellow to dark brown. With vitrinite reflectance value between 0.6 and 1.5, that indicates immaturity and correlates with light yellow color of spore, whereas the vitrinite reflectance value above 3 suggests essentially graphitic condition of organic matter without any hydrocarbon, when spore coloration also turns into black. Now, these values, however, only give idea about degree of maturation and temperature attainment, and they are in no way indicative of hydrocarbon generation propensity of organic matters. Hydrocarbon generation capacity is fully dependent on the type of kerogen and organic matter, that is whether they are humic, sapropelic or the mixed. This criteria, different criteria what we are talking about, it only talks about the maturation of organic matter, not the propensity of the organic matter to yield hydrocarbon. Now, this is a curve that clearly shows the correlation between the different criteria that we talk about the maturation of organic matter. On the left hand side, the black portion that clearly shows the oil window, the upper gray portion that shows the biogenic gas release, and the lower gray portion is the dry gas that we say because of the thermocatalytic action. And you can see what the temperature that it represents the different portions, and the vitrinite reflectance, the spore coloration, the level of organic metamorphism, then hydrocarbon, and what are the hydrocarbon products at different stages, that is diagenesis, metagenesis and catagenesis, that we expect that that is shown in this diagram. Now, whatever we have discussed, if we summarize that depending on mode of origin, that is terrestrial or marine, vascular, plant, algal, spore, pollen, etc., organic matters are classified as humic and sapropelic in character, and further classified into 4 types of kerogen, that is type 1 to type 4, on the basis of their hydrogen is to carbon content. Along with host sediments, organic matters undergo burial as they come under the influence of biogenic and chemical transformations. At shallow depth of burial, that is at the diagenetic stage, organic matters transformed into thermally reactive complex that we call the kerogen, and which is made up of carbon, hydrogen and oxygen with minor amounts of nitrogen and sulfur. Dehydration and decarboxylation are the 2 most important reactions at this stage. Net outcome of all diagenetic reactions is the reduction of oxygen content in the organic matters, keeping carbon is to hydrogen ratio intact. Subsequent burial expose kerogen to high temperature and prompt thermal cracking of complex organic compounds into biomonomers. Formation of oil from suitable organic matters is best noted between temperature 60 degree and 135 degree centigrade. Conventionally, we refer it as oil window. At temperatures more than 150 degree centigrade, thermal cracking of hydrocarbon that starts with formation of methane. Now, beside temperature, the other important factor responsible for maturation is the age of the sediment. A balanced combination of temperature and time can yield the best possible hydrocarbon out of organic matters. So, a number of chemical and biological parameters and indices are devised in order to study the maturation of sediments and their contained organic matters. Amongst these, vitrinite reflectance, pore coloration index, hydrogen index and production index are some of widely used maturation indices. However, it may be noticed that all these parameters and indices, they only talk about the thermal maturity of the sediment and they in no way talk about the hydrocarbon potentiality of the sediment. Hydrocarbon potentiality of the sediment only depends upon the type of organic matter and the total concentration of carbon present within the sediment. I hope you all have enjoyed this module of the petroleum geology. Thank you.